Good morning, my dear students. Most of you, suppose there are 100 students of MBBS, most of you will be going for clinical practice, will take MD in certain specialties, and will do clinical practice. That is a good work. But some of you may be interested in a research career. And therefore the question comes, suppose I enter into a medical career, what will be my prospects? How can I progress? These are the usual questions that is asked. And therefore, I would like, therefore my work is to give an introduction to this part. We shall go for a small presentation. Today's topic is mainly prospects of research career for medical professionals and research and its possibilities for a medical. Now, I would like to go in this fashion that the topic covered. That is, one is need and importance of research in medicine, careers available in medical research or career options, R&D in diagnostic industry, skills needed for a medical researcher, potential employers, and career progression. Most of them will take only 5 minutes. The second and third will take a little more time. Now let us first see what is the need for research in medicine. See, all the medicine that you are practicing now or going to practice later on, that is dependent on the research work that is carried by somebody else. See, for example, if you are using antibiotics today, that is 20 or 30 or 50 years back, somebody has brought out that antibiotics. Let us take two just examples. One is this COVID-19. It is produced by a few months back, that is the December 2019. A new virus that is later on named as SARS-CoV-2, and that is a new virus that is produced in China, and that is now rampant in all over the world. And now, see, you can see that within January 2020, by that time, that is within 2 months or something like that, we get the genetic nature of this particular virus. The exact details of the DNA sequence of that particular virus has been available. How it is possible? Because of the research. And now we are going for the people are working on making some medicines, etc. on that. And therefore, within, let us say, within 1 year, we would like to completely eradicate. Not eradication may not be possible, but sometimes the treatment will be possible. Treatment as well as the vaccines that is going to come, let us say, within 1 year of the starting of the pandemic. Therefore, that is the area where we want new research that is coming up. This is the importance of research, or need of the research in medicine. To take one more example, that you are already familiar with AIDS. That is in 1981 or 82, that is started as a small pandemic like that. And then at that time, nothing was known for this virus. And within 2 years, that particular virus has been isolated. Its genetic features has been identified. And from that onwards, that the real treatment procedures, the medicines have been produced, and because of that, the whole pandemic has been, you cannot say that it is eradicated, but that has been controlled now within, let us say, within 20 or 30 or 40 years. Therefore, that is the need for research. That is new work on these newer things has to be done. Then only we can proceed further in the medical field. In life, new problems, events, and processes occur every day, and solutions are required for tackling the new problems that will coming up. And scientists have to undertake research on them, find their causes, solutions, explanations, and applications. And therefore, that is an important feature. And of course, all of you, all this, let's say there are 100 students in an MBBS class, all of them cannot go for research. But those who are interested can go. A medical scientist, through research, determines the causes of the disease and then develops ways to prevent and treat them. Now, I want to just say approximately 120,000 people were employed per year, that is new recruits. This much is in the US, that is in the medical researchers are taken per year in the US. Of course, in India also may not be that much number, but still we want newer candidates in this field. In other words, that there is a good possibility, there is a good opportunity for a research worker in the medical field. That is only what I want to emphasize at this moment. And this employment will grow faster than the average growth of other occupations. The pros and cons of career in research is that one is that independence, that you have your own priorities, you can organize your own working, your priorities and your schedule, that can be you can choose that. And the prospects that I shall be going greater detail later on, that career prospects, progressing in the postdoctoral position, then to professorship, etc. That of course it is competitive, but at the same time there are good ample opportunities available. Now, the first part is over. The second part is the careers available in medical research or the career options. There are so many career options that you can think of. And these are some of the, this is not a full list. It is only just starting. That you can, any number of, you can increase to 50 or 70 like that, these career options available. The only thing is that there are, let us say, 28 departments in the medical college. All these are opportunities for your career in research. Now, I just thought that the hot topics, that is, I just mentioned stem cell research, molecular biology, etc. Some of these. Again, I shall describe a little more greater detail in these areas. The list of careers available, I just want to impress you on that. That the list of careers available within the sectors of medical research is practically endless. You think of anything, and that is a topic that you can go on. Now, one by one, little more examples we can take. For example, pharmaceutical industry, where a lot of medical people are necessary for the pharmaceutical industry. If you remember that India is one of the foremost pharmaceutical industry in the world, about more than 50% of the total pharmaceutical substances, drugs, are produced in India. And therefore, there is careers in research, drug development, and clinical trials. A lot of medical people are required. Of course, if you want to develop a particular drug against a particular disease, that is a big process. Usually, it will take 7 to 10 years. Usually, suppose there are 100 molecules you are trying that is effective against, let us say, cancer. Then for that you want to do first tissue culture, and then if something out of that 100 molecules, let us say 10 are possible to do, and then you have to do animal experiments, and then perhaps three or four may be useful at that time. Then you go for the clinical trials, human clinical trials, phase 1. Phase 1 means that the toxicity is, what is the toxicity, what is the dose. and that out of that only one, you start with 100 molecules, you get only one molecule at that time. Then go for clinical trial 2, let us say about hundreds of patients are taken, and whether that particular drug is effective. In the case 3, you get more than 500 or 1000 people, and then only you can go for manufacturing and marketing. Therefore, this is a very long-term procedure, and all these areas you want research workers, and therefore that is an ample opportunities are available in the pharmaceutical industry. Of course, biochemistry, since I am in biochemistry, I just wanted to show that study of any form of life, from single cell amoeba to virus to humans, with physiology, and any topic that you think that, you see, in my textbook I usually write that the techniques, all the techniques for the research is biochemical in nature. Therefore, you start with anything, you end up in biochemistry. That is what I usually tell to students. There is another big opportunity is in the epidemiologist. They sometimes tell that this is the disease detectives. They investigate the cause of the disease and help to control their spread. They look for patterns, trends, and commonalities between people who have the same disease, investigating where it started and how to stop it spreading. And this is a highly rewarding job. If you remember historically, that is very interesting that John Snow originally started with that there was some epidemic of cholera in England, a particular area. And he showed that all the persons of that particular cholera epidemic has been started with one particular pump, that is water pump. That pump water has been contaminated by the cholera virus. That pump is still remaining in England. That is for a study of contaminated water as the likely source of cholera. That has started. It is in 1854. He described that, and that is the perhaps one of the very daring opportunities of the research work in this particular field. Another comparatively recent one is that the Burkitt lymphoma. Dennis Burkitt was a general practitioner in Uganda area, and he seen that a particular type of disease that is coming in that particular area. Originally he thought it is a sarcoma. He told that a particular type of sarcoma is available in the African population, only in a certain particular locality. And that is why the epidemiology comes, that he showed that he pinpointed all these incidences and showed that it is only a particular area where there is a particular type of rainfall and particular type of temperature, and only in that area this particular disease is available. And therefore later on that particular Epstein has identified that particular virus in that lymphoma series. Of course, Epstein himself is told that it is not a sarcoma, but it is a lymphoma. And anyway, these are some of the areas where this is the first area, first virus that has been identified as an oncogenic virus in human beings, and that is the historical importance of this particular work. And this has been done by only a very few people with very little research inputs, that is the particular funding. With very little funding they had done that a particular historically very important research work they had done, and that is the epidemiological work. Then another example of epidemiology is that the famous Framingham heart study started in 1948 and still continues till date. That is the first evidence-based clinical study for heart disease, most well-planned medical research in the area of heart disease. Epidemiology of risk factors of cardiovascular diseases. That is, the study was started about 5,000 people at that time, and about the 30 to 60 years of age group that they picked up And at that, those don't have any heart disease at that particular moment at the entry level. And then, of course, it has been increased. Now it is first generation is over, second generation. Now it is third or fourth generation of participants are the total 14,000 participants. The work is still going on. And the classical example of identifying the disease, the areas where the disease, the heart disease is prominent, what are the causes, the effect of hypertension, diet, exercises, blood, cholesterol, and other parameters have been identified, and that is classical now. And therefore, this is the epidemiology, the importance of epidemiology that I just want to emphasize at this moment. Then there is the clinical immunology, the study of how pathogens affect the immune system. And immunology is one of the most rapidly advancing areas in the biomedical research. It contributes to the eradication of infectious disease, as well as the development of successful strategies for vaccination and also for organ transplantation. And these are the cutting edge areas of the research work now going on. Joseph Murray got Nobel Prize in 1990 for the first kidney transplantation in 1954. And the kidney transplantation, organ transplantation has been an important area of the research work still going on. A little away from that is the so-called monoclonal antibodies. Now you can think of, see, almost all antibodies that you are working are now becoming are monoclonal antibodies. This is not the time to go how it is produced, etc. Only to show that these are the anything that any antigen you think about that you can produce now monoclonal antibodies. Now in the area of clinical, say for example in the cancer treatment, there are so many antibodies that has come, and that has been most of these monoclonal antibodies of active only at specific particular area of the pathway, and therefore there are so many pathways that can be arranged, that can be thought of in tackling cancer. And therefore, so many antibodies has already come up in the clinical practice. Another big area that you can think of as a clinical person for research is the nutrition and dietetics. Food safety, microbiology, preservation, and food production. These are great areas of the human consumption. We ensure that food produced meets all the necessary standards. They are also concerned with finding the best ways to convert the raw ingredients into food products. Let me take that last one, that food industry employs up to 400,000 people. This is per year and accounts for over 15% of UK's manufacturing output each year. And that is going on increasing. That is a big industry that is coming up. And therefore it needs a lot of persons for research in that particular area. The nutrition, another important thing I wanted to tell that nowadays this is, see, the research is important for the total quantity of food that is available for the human beings. When we were students, that is 1950s, etc. In that area, India was facing famine. The people were thinking that India is doomed because of the famine. But that has never occurred because the nutritionists and agriculturists, they thought for producing newer forms of substances, and therefore the famine was never came. Now, of course, in the future, it is again coming up because population is increasing, but we have already reached the potential of the agriculture. And therefore, this is the time that people are going for manufacturing food products, that is proteins and carbohydrates, by the recombinant DNA technology. But that is a great avenue that is coming up in the near future or in the 10 or 15 years now. That is DNA, recombinant DNA technology that you are already aware that any DNA from any substance, a useful DNA can be transformed and added to a plasmid, and that can be introduced to the bacteria, and that bacteria can go on working, and therefore any number of that particular substance can be produced. The best example is the insulin production. That human insulin can be produced by this method. That is, the bacteria can produce any number of insulin nowadays, and therefore that is the advantage for DNA technology, and therefore that is a growing technology, that is a growing area, and newer and newer products are to be produced, and this needs the researchers, and therefore you are welcome in such areas. Insulin food technology I had already. Of course, the future is gene therapy. Now, I'm not going in greater detail. It is in all the textbooks. You will give more details. That almost, the single genes that is available, for example, phenylketonuria, such things that can be tackled by gene therapy. That is, the correct gene can be introduced to the patient, and therefore that particular disease can be eliminated, and therefore a good person, a useful citizen can be produced. And in that respect comes the stem cells. This is an advancing technology that is in the very cutting edge technology that is now coming up. The hematopoietic stem cells, mesenchymal stem cells, tissue specific stem cells are now available in these are more and more work is going on. The last few years, the Nobel Prizes have gone into people working on these stem cells. The therapeutic potential of mesenchymal stem cells are many. Just two examples. One is the tissue repair and regeneration, immune suppression. This has already come into the clinical field. Cord blood mesenchymal stem cells are very useful for taking the stem cells. Umbilical cord blood can be taken. In our own research, that umbilical cord is being used, that is for the work. See, these are the various potentialities for the cultured pluripotent stem cells, starting from the right side, the gene therapy, the liver cells for skin and tissue, say burns. We have a very big burn unit over here, and it is also useful for liver failure, burns, bone trauma, pancreatic islet cells for diabetes. This is a very promising area where diabetes can be really treated and cured by introducing the pluripotent stem cells. Similarly, heart cells, the cardiac disease, that the area of heart has been is killed by the, and now that heart muscle can be regenerated by giving the stem cells. The nerve cells. See, that nowadays the telling is that when you have a cut of a particular nerve cell, then there is no more. The nerve cannot further progress. But people have already shown that if you inject these particular stem cells, that particular nerve cell can regenerate. That is already shown in the laboratories as well as in animal being, in animals. And therefore, these are now potentially that is useful for Parkinson's disease and Alzheimer's disease. Similarly, bone marrow and leukemia therapy, that you will study that in greater detail later on, that has already come in the clinical practice. Skin and tissue regeneration for burn victims, diabetes, spinal cord injuries, I have already described. That is the upper side. See, these are the blood glucose level and transplantation that the stem cells are very useful for for treating and curing the diabetes. See, we generally say that cure is not possible for diabetes, but here is a possibility by using these things, and therefore people are very much interested nowadays in the stem cell work. and you are also welcome in these slides. Now, these are the clinical trials that are going on in India on the these stem cells. That is autologous bone marrow derived cells for necrosis of femur head, type 2 diabetes mellitus, optic nerve injury, management of facial nerve paralysis, myocardial infarction, and also patients with acute ischemia stroke. These are the areas in India. These are going on. Therefore, there is great potentialities for this particular type of work. The stem cell architect is Martin Evans. He got Nobel Prize in 2007. Similarly, it is also a DNA repair mechanism, Paul Modric Nobel Prize 2015. These are very much related with these type of works. See, Elizabeth Blackburn, that is the telomerase. Telomerase is the enzyme that is necessary for the total correct replication of the DNA, the total replication. If the telomerase is not there, the final areas of the gene will be deleted. And the telomerase, for example, we are in our own institution. There is a good there is a group of people working on telomerase. That is why I just wanted to took this particular place. Similarly, that is related with Venkatraman Ramakrishnan. He is now got Nobel Prize in 2019. That is also related with this. Another area which is very much growing nowadays is the medical informatics. It is said that there is 21% growth rate in medical informatics, whereas in the average medical research there is only 7% interest growth. And therefore, this is an area where you can certainly think of, and that is this almost 50,000 jobs every year that will come up. That is, they need healthcare, technology, and business informatics will combine all these activities. And specializing in areas like bioinformatics, medical informatics, and public health, this will come. Now, perhaps the world is moving towards artificial intelligence, and accordingly, medical field also has to be taken into that consideration. This refers to the stimulation of human intelligence in machines that are programmed to think like humans and mimic their actions. More and more artificial intelligence is coming nowadays. For example, there are the surgery. There are so many surgical instruments that are available, that is artificial surgeries going on, that is available in our own centers. In Kerala itself, that is already has started. And say, for example, my own one of my students, he has taken up a program that is this is US grant is available for that. 30,000 cardiac infarction patients, he is taking blood from them, 5 ml blood from them. 10,000 normal persons as controls, and in each of them he is he is studying what happens in 1,000 molecules in blood. In blood, there are so many substances. He is taking 1,000 molecules in each patient, and that is by mass spectroscopy. That is now available. That is now possible. Therefore, if you take 40,000 persons into 1,000 molecules, that will come 4 crores of results. Will come within that will come within 1 or 2 years. Therefore, this much result you cannot go through by normal human beings. Therefore, it needs artificial intelligence. Therefore, these are the areas where people are going on more and more on research. And this is the ultimate aim, is that what is the difference between the cardiac infarction patient and the normal patient? Is there any difference? Any marker is available out of this 1,000 molecules that they are studying? Is there any marker? Is there any special molecule that is present only in cardiac infarction patients, not in normal persons? But these are the ways that people are going on. Now, I just wanted to go to the third thing, that is the research in diagnostic field. When you are studying biochemistry, you are going for its theoretical aspect as well as some practical thing. But you are not very much familiar with the clinical biochemistry. Of course, you must have gone to our laboratory, etc. But that is one of the most important areas of biochemistry. The R&D in the in vitro diagnostic industry is very much important. In vitro diagnostic industry is a part of the big pharmaceutical industry. But at the same time, it is very vital for the doctors. See, the International Federation of Clinical Biochemistry, that has come up recently after a very big study, that about 75% of the inputs that is necessary for a doctor to get a diagnosis, about 75% of that input is coming from laboratories. That is by the biochemistry, not only biochemistry, microbiology and other X-ray and all other imageology techniques. All these will come there. But what I want to tell that this in vitro diagnostic industry has become a very big challenging area for the medical field. The organizations must adapt their products and services in order to remain competitive. Otherwise, they will lose revenue and fall behind the market. Therefore, this is a very competitive field, and there needs a lot of medical people to come up with this type of research. I just take one example. So there is something called the turbidimetry. Turbidimetry, you are familiar with the colorimetry. Colorimeter, one color is produced, and that color intensity is identified as quantitative. Here, turbidimetry means that you are purposely making the solution as turbid. That antibody is added, specific antibody is added, and that particular protein is precipitated. A turbid solution is produced. Light is passed through, that light is scattered, and what is coming out, that will be identified, that is quantitated, and that is and the absorption of that light will be proportional to the amount of that particular protein present in that solution. And this is the advantage, is that it is a one-step procedure, fast, cheap, and quantitation is possible, and most versatile thing. And this is the turbidimetry technique, that the light source is there, sample is there, and therefore the transmitted light, that is the light passing through 180 degree, that is you are identifying. On the other hand, there is something called a nephelometry, where the same sample is put, and the light is scattered, and light is passing deflected 90 degree, and that is detected, and that is taken as the for quantitation. And that is therefore people were working on this nephelometry and turbidimetry for a long time. The turbidimetry and nephelometry were useful, but then we thought, some of us thought, that why not you combine these two things together in one particular instrument. All the last 40 or 60 or 70 years, there were nephelometry instruments and turbidimetry instruments for the clinical laboratories. But now we brought these two together. Therefore, the light is passing through 180 degree as well as to 90 degree. Both can be identified, both can be quantitated. And that is the turbidimetry was used to determine proteins present with high concentrations, whereas nephelometry is used to determine proteins with the lower concentrations. That is the difference. However, nephelometry and both the turbidimetry, these instruments were very costly. One particular nephelometry will cost about 50 lakhs of rupees, and therefore that is not possible for an ordinary laboratory, a clinical laboratory, to purchase it. Nephelometry was not popular because of the cost of the instruments. Therefore, we combined these together. That is the MISPA I2. That is the about 7 years back. It has been in market. MISPA I3. That is the further development of that. That has come in 2 or 3 years back. About 60,000 machines have already been sold in the whole world. This is one of the first time in the whole world that this particular newer technique has come. It is a serum protein analyzer. Both nephelometry and turbidimetry is identified. And the advantage is that automatic selection is possible because machine itself will identify which particular technique has to be used. If the protein quantitation is high, and then it will go automatically for turbidimetry. Protein concentration is low, then it will go for nephelometry. That automatic selection is available. Cost is given 1/10 of the cost of the of the big institutions. That has been come down to 1/10 of the price. 25 different protein parameters and newer parameters you can add on that later on. A lot of awards have been brought on that. This is one such award ceremony. This is another award ceremony. Another award ceremony. It goes on like that. And it has been identified by the Department of Science and Industrial Research. 4 patents are already there. And therefore it is a big well, this is only just there is something wrong in this one. What happens to COVID? The COVID, that the virus could be identified nowadays by the PCR technique. That is the standard technique that is available. But the difficulty is that the technique needs about 5, 6 or 7 hours for that particular work, and therefore usually you will get the result on the next day, whereas the patient needs the result immediately. Therefore, we tried in the few of our students have gone for a newer technique, comparatively new technique. Instead of PCR technique, they are going for the LAMP technique. LAMP, the full version is that reverse transcription loop-mediated isothermal amplification. The difference is that primers are slightly different, that you have to develop yourself. That is the intellectual area, that the primer development. And then if you have that, then the LAMP technology carries the reaction at a constant temperature. That is the advantage. Whereas in the usual reaction, that is the RT-PCR technique, that reaction is taking place in 3 different temperatures, where you need a particular instrument for that temp cycler, that is called, and that is costly, and that is required, that it takes a lot of work there, and therefore there is more chance of certain negative results. This is becomes very economical and quicker, and it is 100% acquired. The advantage is that the actual LAMP technique will need only 10 minutes of work, and therefore the total test time is shortened to 1 hour. But this is a great leap in that particular area, in the clinical laboratory technique. This is the RNA is extracted, reverse transcriptase is done, and cDNA is produced. The LAMP technique is that only needs 10 minutes, and a dye is added and fluorescence is detected. But that is very that the positive or negative, that the particular patient is positive or negative, can be identified within 1 hour, and the particular test has already been sent to ICMR for the validation, and I do hope that within 1 or 2 weeks that will come in market. But this is the area. I just want to give some examples, that is all. And another area is the microarray technique. One of my students is doing this particular work, and that DNA probes can be done. 500 to 5,000 genes can be put together in a particular well, and that can be identified, hybridized, and that can be identified. Is something like that. The altered normal cell and a cancer cell can be identified, and you will get something like this. But that red areas are the genes which are amplified and the green areas are the genes that are suppressed. And this micro application is very big, that tumor diagnostics, new drugs development, infectious disease diagnostics, allergy diagnostics, all these are possible and therefore welcome to these fields. And the thing is that the next is, I don't know, this is not working properly. These are the skills needed for a medical researcher. Of course, you need a medical degree, and then you can go for PhD or specialization as a medical thing. And perform, you should have laboratory work and also learn about the research methods. These are time consuming. Of course, it will take any doctor for that matter. If you want to come see after the MBBS, you need to go MD for a specialization, then you go for, it will take at least minimum 10 or 15 years to get some level. The same thing will take here also. You need a PhD and then go for laboratory work, and then a career is developed. That will take about 10 or 15 years to get at top level. What soft skills you need? That one is critical thinking, writing and verbal communication ability, reading comprehension to see what others have already worked in that, problem solving or analytical thinking, and ability to work well in teams. That is, a teamwork is very important in research. Of course, if you are going a pure clinical practice in your own practice, then you are alone. There is no need for this particular skill. Whereas in the research nowadays, that there is usually about hundreds of people are working together. And therefore, unless you can fit well in a teamwork, you cannot progress well. The attributes, this is the same thing, but just only two things I just want to emphasize. One is dedication. That is, your work, you have to be concentrating, you should be very persistent on that. That is, there will be setbacks. See, research means that you cannot say you are not sure what is happening. You may or may not get the result. Therefore, there is a good possibility that 50% chances are that you will not get a result. Therefore, in that case, you have to repeat it and change it. And therefore, that is what I say, persistence. That is an important attribute for a research scholar. Of course, what are the responsibilities? That you have to plan and do the work and interpret. Very important is keep accurate records of the work undertaken. See, after 10 or 15 years, somebody will question you, and therefore at that moment you have to bring back your records and say that I have done this. And if you don't have the record, it is assumed that you have not done that work. It is very well, it is in the court of law also, it is something like that. If you have not recorded that in your clinical details, that the court will think that you have not done that work. Similarly, it is happening here also. Therefore, keeping the record, accurate record, very meticulous record is very important for a research worker. And of course, you have to get funding, you have to attract funding, and that is, you have to work for that. And collaborate with industry and other research institutions. The employees will expect certain things, that you are highly motivated, you should be a highly motivated person. There should be flexibility and creativity, capable of technical writing skills, and sound scientific clinical judgment. One question I want to ask you, what was Christopher Columbus' greatest achievement in discovering America? One answer, generally speaking, is that he took advantage of the spherical shape of the earth to get into India by the western route. Yes, but many people before that also knew that the world is in a spherical shape, and that the idea occurred to others also. People were trying on that. And the second answer is that therefore that is not his unique achievement. He prepared his expedition meticulously and rigged his ships most expertly, etc. That is also many other persons have done previously as well as after that. But the most remarkable achievement was that he took a decision to leave the known regions of the world and sail far beyond the point from which he could get necessary provisions. That is the the thing that he has done. In science too, it is impossible to open up a new territory unless one is prepared to leave the safe anchorage of established doctrine. Therefore, go beyond what is now available, and therefore you go far away from what is now available. That is the necessary, that is the challenge of that is research. And all these topics I have covered. The last one, I think this is the. You may think, what are the areas where you can get employment? This is one is, of course, Indian Council of Medical Research. That is perhaps the best option. Then there are so many clinical research organizations are available. Pharmaceuticals and biotechnology companies are now so many in India. As I have told that India is the biggest area where pharmaceutical companies are available. National health related charities, private hospitals and trusts, universities and science centers. You know that even here there is a small group of research workers, and therefore that is growing on. The potential market is increasing. Indian Council of Medical Research, they have their own national institutes. So many, about 20 national institutes they have. As Indian Institute of Cytology, Indian Institute of Virology, etc. There are so many national institutes under them, and there are extramural research grants are available. As Dr. Varghese has told, that there is so many types of fellowships are the short-term fellowship students. You also must have already got, some of you must have got short-term visiting fellowships, that with this that you can improve your techniques and go somewhere else. Department of Science and Technology, Government of India is another big organization that need doctors as researchers. See, there are more than 50 scientific institutions under science and technology. Just two examples in Kerala, Sri Chitra Institute and Rajiv Gandhi Center of Biotechnology. These are under Government of India. And then there are so many Indian Immunology Institute, New Delhi, Tata Memorial Cancer Center, Mumbai, Indian Institute of Science, Bangalore, Center for Cellular and Molecular Biology, Hyderabad. There are so many institutions, and these all need newer researchers, and therefore the opportunities are ample, and therefore only you want to start your research career. And they have also their research projects, various types of researchers, and young scientists, they have a fast track scheme, and young women scientists scheme, there is a special schemes are there for them. And now the last topic, the career progression. The key, how you are entering into the research field. Now, what will happen to me? That is a real question. Key to career progression is publishing. That you do research and publish, and depending on that, you can your career will be benefited. Career development tends to be more structured in industry rather than in pure research institutions. That is all. Just one example I want to say. This is Soumya Swaminathan, born 1959, MBBS from Armed Forces Medical Sciences, MD from All India Institute. She was director of National Institute of Tuberculosis, Chennai for about 10 years. Then became Director General of ICMR for 2 years. Then Deputy Director General of WHO for 2 years. That was, see, in the WHO, about 10 Deputy Directors. She was one among them. Now she has still improved that she is Chief Scientist of WHO. So from 2019 onwards, that is supposed to be the second in command. That is most probably she will become the head, the Director of WHO. I just want to say that she just opted the research after MBBS, etc. She just opted MBBS and ended in the National Institute of Tuberculosis as a small research worker. Like that, the career progression is possible. That is another just example. You must have already seen him perhaps. He is my student. About 25 years back, he was MBBS student in Thrissur Medical College. Now he is co-director, Institute of Human Virology. He is second in command in that institution. That is one of the biggest. You must have heard of Robert Gallo, who is the founder, who identified the HIV virus, and that he is the Robert Gallo is the head, and this man is the second in command of that institution. And therefore, just I want to emphasize that there is plenty of opportunities, and you can go through any your career opportunity. That your progression is also very good in this particular field. With that, I remain. Thank you.